Hello and welcome to ChatGCP, a medical science, clinical research and commercialization podcast, and I'm your host, Charles Beasley. As medical technology advances, we can expect genetic data to be used more frequently in both research and clinical settings. The promise of precision medicine is that we could use our unique genetic fingerprints to inform healthcare decisions, and thus get the most out of our health, whether it be treatment, prevention, or longevity. As more people have their DNA sequenced, it is critical that this information is kept secure. We're all aware of the importance of protecting our personal information. You wouldn't share your home address or bank account details with the public, for example. And by virtue of its unique fingerprint, genetic data is the pinnacle of personal information. It is unique to that individual, and it is also enduring. In today's episode, you can expect to learn the importance of genetic data in the future of medicine, how genetic data could be used by both benevolent and malevolent parties, and some of the challenges that need to be overcome by founders developing medical technologies for the future. Here to unpack these topics and more is today's guest, Kris Vette, the founder and CEO of GeneCrypt.
Kris, welcome to ChatGCP. Great to have you on the podcast today.
Great. Thanks very much, Charles. Great to be here.
Awesome. So you are currently the founder and CEO of GeneCrypt, a startup company providing end-to-end encryption of genomic data.
Correct.
That sounds like a bit of a word salad when someone like myself, uninitiated, says it. But can you describe for us why it is important to have such a service as an end-to-end encryption of genomic data, and how did you arrive at the importance of such a startup?
Sure. So probably the way to answer that is to take it back to where medicine began, which was probably medicine 1.0 back in Socrates' time, and bring it forward. So, we saw the evolution of medicine around the hospitals, physicians, surgeons, family healthcare physicians, GPs as they're called here. And that really played out over the last few hundred years or 1,000 years. More recently, since the introduction of antibiotics, we dealt with infectious diseases, and more recently in the last probably 70 years we've been dealing with chronic disease. In 1953, Crick and Watson broke down DNA and uncovered the code to life, and more recently we've completely decrypted the human genome. I mean, as much of it as we think we understand at the moment. Genomics is basically DNA, or the code of DNA, and how we secure that into a file format.
Yep.
And more critically, we're starting to understand that genomics is the blueprint, and it expresses the proteins and therefore and metabolites in the human body. So it is critical that we each have a way to grasp what our own individual genome is, and then as medicine plays out into precision medicine, we'll get into that later, we need to have a reference point to go back to in order to compare what our particular DNA is as an individual, because we're all different. Everyone's got different DNA. It's highly related to our inheritance from mother and father, but fundamentally it is unique to us. So it's highly identifiable and therefore highly sensitive.
Yeah, so it's a matter of keeping that unique signature of one's DNA secure and preventing it from being accessed by third parties, which I'm sure a lot of them have very, high-minded, important things to do, but there are some bad players out there that would use your data, just like any of your personal data, for nefarious purposes. So essentially, it's the importance of securing your personal data. I mean, when you sign up to a website, you're all about, there's VPNs for everything to secure information. So this is an extension of that, essentially, personalized information, keeping that secure.
Yes, that's a good summary. Fundamentally, humans need access to this data because as a human race we need to progress medicine, so we need access to the value of that data, but we need to protect our individual data. So it's a two-sided coin. There's value in it, but there's also danger in it with loss of personal control or loss of security over that data.
And I suppose with every new technology, there are known unknowns and there's unknown unknowns, and this technology is kind of preempting both the known unknowns and the unknown unknowns. Like, we don't know who's going to use this genetic data, what it could be used for, good or bad, but let's put the safeguards in place to ensure that we have good access to it, and yet it's secure and safe and retrievable, encrypted, right?
Yes, yeah, absolutely. I mean, I think, you know, you can play out the science fiction of it, or even, you know, verging on reality, the James Bond films with weaponizing DNA, and there is potential for that. If you wanted to identify specific variants that were, or a specific set of variants that were unique to one particular family or one particular race, there is a theoretical way to weaponize that. There are, and that played out in one of the James Bond films. I think they got it wrong. But from a more realistic perspective today, there are highly identifiable ways to use that DNA to both track someone or to fingerprint them effectively. It's a fingerprint of an individual, and also to predict the future of that person's health. I mean, it's not usually one-to-one prediction. It's often a risk or a risk score, and certain genes are highly predictive of a future. I mean, a Huntington's gene, for example, can be quite malevolent in its prediction. But still, you don't want employers or insurance companies working out your potential future based on what is just a risk score, and then using that potentially against you in terms of how the costings work, or potential job and employment prospects work.
Right, so it could be potentially used as like a genetic minority report.
Yeah, absolutely, absolutely. And there's
play out different circumstances.
Yeah, very, very much. And so we've got to be careful of prejudicing against specific groups of people and inheritance. There are strong inheritance lines through, Pacifica or Maori, other indigenous groups. In the case of the BRCA gene, there is an inheritance line through Ashkenazian Jews, and so you can quite easily become prejudiced in a way that is unfair, because that predictive ability is only a risk score, and someone's lifestyle may well work over the top of their genetic inheritance. And so a little bit of knowledge can be a dangerous thing. You don't want such sensitive and individual identification of data in the public domain.
Yeah. It's almost like the argument of determinism. It's genetic determinism. Yes, there are some. It's a range, right? It's a spectrum. Some are highly predictive. You brought up Huntington's, which you only need one gene, Huntington gene, to have a risk for. Well, you will develop it; it's guaranteed. But at the timing of which is dependent on the unique aspect of the gene. But for other things, I've heard stories where some people can have a genetic predisposition to developing lung cancer. More than others, and to the point that you can be a chronic smoker and never develop lung cancer. So it's highly complex, and if you're not initiated in the art of genomics or bioinformatics, like, you could get it wrong. It could be used against you, like you say. And we're very important about not discriminating according to other personal data like name, culture, location, place, country of origin, that kind of thing. And if genetic data is just an extension of that, it also needs protecting from discrimination.
Yes, yeah. I mean, to be clear, I'm not a clinician and I'm not a geneticist. My original undergraduate training was in neurophysiology, so probably relatively far removed from genomics. So I don't pretend to be an expert on the specifics of the genetic disease. But clearly what you've said is correct, to the point where, the inheritance patterns and certain genes, I mean, you brought up Huntington's, which can have usually can have a nasty long-term outcome. And so it's important, A, that people aren't prejudiced against, and B, if that data is out there, or if it's been used, if someone has had a whole genome sequence done, playing out, say, a neonate who has got a particular syndrome that needs to be identified early for intervention with a specific drug or metabolite that might, in say 15 or 20% of the cases, turn the disease around. So they might need early whole genome sequencing, which is much more predictive and has a higher diagnostic yield than the current testing that's done. But if that potentially exposes a future, Huntington's, or even an ApoE4, gene, which has something like 15 to 25% of the population have that gene. It's involved with lipid metabolism, but it gives an increased risk of dementia. It doesn't mean you're going to get dementia, and there's probably a lot of lifestyle factors that can override that. But if you were going for a job interview at age, as a CEO at age 40, for example, you wouldn't want your employer knowing that you had an ApoE4 gene. I mean, in case they might not want to take that risk, and yet it may be totally irrelevant to your future. So there are valid reasons why genetic testing for diagnostic purposes, even removing the potential for predictive and preventative reasons, which we can maybe come on to later around personalised medicine. But there are very strong reasons why, say, in early childhood or at any stage in development, you don't want everyone knowing what that read is. In fact, you may not even want to know yourself. If one of your parents has a BRCA gene, for instance, which has a relatively strong inheritance, or Huntington's, the offspring of that person need to make their own decision about whether or not they want to know that, and they can through our technology we can mask that out based on their permissioning of the gene regions they want access to, or they want their clinical team to access rather.
Yeah, that ApoE4, is that the Chris Hemsworth gene?
I think so. I can't.
Because that's the one that I think he was on a show recently with Peter Attia, and he got told he had some gene that increased his risk for dementia.
Yes, I'm not certain if it was ApoE4, because ApoE4 is relatively common, but I think he was showing some symptoms. Right. I believe he disclosed he was showing some early symptoms already.
I see. Because I can imagine that if you get told of your genetic status and certain things, and you can't do anything about it right now, based on current technology, that's not very helpful. In fact, it's probably very detrimental to your mental health. Yes. And as we know, we've come more and more to discover that your physical health and mental health are intimately connected. So that's not a good outcome for anyone in it.
Yes, that's the ethical dilemma with something like Huntington's. I mean, as we increase our ability, actually, there's a good example at Auckland Hospital. We've got a kind of world-leading professor there. I haven't met him before, Professor Ed Gain. He's recently discovered a cure or been involved in a clinical trial using CRISPR, gene editing technology, for a familial amyloidosis, which is a rare disease but usually fatal. And it looks like him and his team, as part of a CRISPR leading-edge study, have potentially cured that. So that is an example of, if you had inherited familial amyloidosis, you might now be quite keen to know early on if you've got it, and have the early CRISPR intervention once it becomes once it comes onto the clinical space. So that's an example where we do want to be able to intervene early. A BRCA gene is another one. So BRCA is often the gene that is associated with Angela Jolie. She chose to have preventative mastectomy, double mastectomy, because of, I don't know the exact figures, but it's something like, and there's two BRCA genes. In fact, they're protective genes. So it's if the BRCA is not working, that it has a negative outcome. But she chose to have preventative mastectomy because of the potential risk of, I don't know what it is, maybe say it's 60% risk of developing, an ovarian or a breast cancer by age 50 or 40 or whatever. So in that case, there was something she could do. The other options would be, really aggressive surveillance. So I think in those, and actually in the diagnostic and treatment space, we're increasingly, and that's the essence of precision medicine, which is often talked about as medicine 3.0. Yeah. As individuals, when we've got a cancer diagnosis, we used to hit it, with shotgun chemotherapy, but now if that BRCA gene is identified, there are specific therapeutics that are able to target that, like the PARP inhibitors. So in that case, it's useful to know that you've either got a tumour BRCA mutation, or preventatively, if you've got a germline, meaning an inherited BRCA, you may then choose to increase surveillance. Right. So, we're getting into the weeds a little bit, but those are some of the higher level considerations that I think the healthcare systems need to start thinking through as we move from medicine kind of 2.0, which is about chronic disease management, into preventative and targeted therapeutics.
Yeah, I heard a quote the other day that we're moving, since the advent of kind of industrialization, we used to die, prior to that, we used to die really quickly, accidents or infections that just went systemic and then boom, we're out, into what I imagine is medicine 2.0, which is, like you said, chronic diseases where we die very slowly. Yes. And it would be very helpful to have information that could inform us as to how to avoid that or prevent it altogether. We don't currently have that information, or it's coming in dribs and drabs, with new genomic sequencing or understanding of molecular biology. Whereas medicine 3.0, that is kind of the next generation where we have an abundance of information that can inform us on lifestyle, treatment, prevention, longevity, all together. And that's fundamentally baked into both our lifestyle but our genetic information, right? Yeah.
Yeah, absolutely. I think a good example, you raised it, the one about smoking. So there's I think there's basically a quadrant of genomic inheritance, and so if you happen to be in one of those quadrants, that gives you a predisposition towards inflammatory lung disease, or a significant inflammatory response to a irritant like a smoke, a cigarette smoke particle, you will develop very early emphysema or COPD and go downhill, and you see these people have really poor outcomes. And this is why sometimes you see people are still smoking at age 100. I'm not advocating that, but there are certain people who are protected against that. And so no one wants to, and you would never encourage smoking, but it would be certainly worthwhile knowing that you were going to have a really bad outcome if you did smoke.
Yeah, absolutely. So I think we've really hashed out why it's important to have genetic information for prevention, treatment selection, but also the potential consequences of having that information publicly available or available prematurely when we don't have anything that we can do about it.
Yes.
So why don't we talk about why is it genetic information is kind of higher risk than other personal information? We've kind of touched on it already, but I think you've mentioned in a talk that I've seen you talk about how it's persistent, whereas all other information about an individual, that can change. You can change your name, you can change your sex, you can, whereas genes don't change, right? They are enduring, they stay there forever, and are potentially highly predictive.
Yes. So so outcome data, or not so much outcome data, but phenomic data meaning the data that your expressed physical self. That is typically what it has been captured in medical records, and increasingly in electronic medical records. And this is really goes back to the genesis of where GeneCrypt came from, thinking way back to my time at St George's Hospital in London, where I was managing infectious diseases. So while the data that's collected in typical biomarker data within our health records can be used, it is sensitive information. At one point, I was managing infectious diseases and HIV medicine and genitourinary medicine. So those have quite sensitive data sets within them. But particularly when HIV was a new disease in the late 90s, it was newer for a couple of decades before that. But at the stage before they had brought in therapeutics that worked against HIV, it was a prejudice disease, and in fact, if patients were being seen in genitourinary clinics, sexual health clinics, but also needed inpatient hospital care, they often wanted to mask out their HIV data or their sexual health data. So when we put in the first, I project managed that first electronic health record into St George's Hospital in the early 2000s. So we needed a way to have a separation between the sexual health information and the inpatient hospital information for the HIV patients. And this break glass concept of certain highly sensitive pieces of information being separated off from the typical data set that was available to any treating clinician, and only accessible by specified personal treating physicians for that particular patient.
Right.
And this is where this concept of what is sensitive information, that was before before we were using genomics, increasingly, so there's no doubt medical data is sensitive, but genomic data is a lot more sensitive, because you mentioned it before, you can maybe adjust your blood pressure, or you can live with HIV successfully now. You can change your sex, even you can change your name and your NHI number, but you can't change your genomic inheritance. And that is why it is persistent, and that is why it's sensitive, because it predicts a lot about us. But not only that, it predicts a lot about our offspring and about our siblings and about our parents.
Right, so there's this intricate web of prediction that could be used from potentially just one person's genomic information. You
can tell a lot about identification and about connectedness.
It's quite a nice segue to the risks of this genomic information being used outside of the intended settings, which is in medicine. I think you've already mentioned how insurance companies could use this to kind of stratify people according to their risk, which may or may not play out based on lifestyle. It's all very complicated. But like policing, identity theft, there's a whole host of different risks that could, either now or in the future, play a part.
Yes, indeed. And so this comes back to the importance around the value of genomic data. So it's incredibly valuable for the future of human evolution and development and medicine and drug development, and the kind of work that Sarah's doing. I know Sarah's incredibly concerned about confidentiality around the data sets that they're using to protect the data subjects, obviously, and also there is a concern for collaborative researchers. If they're sharing big data lakes, they may want to protect their own subject data within that data lake, or indeed their own individual researcher pipelines, which may have a high degree of IP attached to them. So there is a way that we've developed whereby you can use confidential inquiry into that data set without disclosing the data subjects or the researcher pipeline. So that is why we need to. That's effectively the main reason why we need to protect those data sets, and we also need to develop a way to allow identity and access management that is permissioned by the data subject owner. Rather, that is my view, and I think this is an emerging view in the world, that control over one's own genomic data set should be ultimately in your own hands. Mm-hmm. The problem with healthcare systems and the way that medicine and healthcare has evolved, only within the last 2 decades have we moved into electronic health records, and they're not even used every day, and they're not particularly secure. We're all aware of the massive data breaches that go on every week around the world. New Zealand not excluded. And so with genomic data, we need, for medicine 3.0, or sometimes predictive precision medicine, we need a way to absolutely ensure that data isn't lost, because once a hack occurs and that data is breached, it's gone forever. You can't retrieve it. I mean, even if you can retrieve it, it doesn't matter because it's already out there.
And it doesn't change over time.
It doesn't change.
So unique fingerprint.
You can't change it.
Won't change until infinity, right? So how is genomic data currently protected? I mean, obviously what you're doing is really pushing the boundaries in terms of encryption and the importance of making sure that it's secure. One would assume that there are current legislative things in place that would protect genomic data.
Yeah, I mean it's loose and it changes around the world depending on which jurisdiction you're in. We know people are becoming more and more aware of this, so there is a problem around patients releasing data for research, or even getting their genomic sequence done, because currently in New Zealand, if you have a genomic sequence done, even if it's done in the research setting, if you are asked by your insurance company, have you had a genomic sequence done, or even a 23andMe, I think they can ask. I don't know that they always ask, but if they ask you and you declare that you have had genomic sequencing done, then they then can choose to alter the premiums or decline you or whatever. I mean, to my knowledge, there are not protections around that in New Zealand. There is a moratorium in Australia for 5 years on use of genomic data in a prejudicial way for insurance or any other reason. In the US, there are stronger regulations, GINA and the HIPAA Act. GINA particularly prevents discrimination based on genetics. Yep. And certainly in the UK, we've got, while it's not specific to genomics, GDPR has very high levels of personal data protection and consequences for breach. Those things aren't so prevalent in New Zealand.
Right, so a bit behind the eight ball down this part of the world.
I think so.
Yeah. And what one could argue that with the potential misuse of that data that is enduring, you cannot divorce it from the individual. I mean, that is information about the individual that will endure for all of time. Really important that we do lock that up carefully, right? I mean, it's not home address, you can move your house. It's not marital status, you can separate.
Yes.
This is you. So it almost feels as though we're behind the eight ball a little bit in this regard, but also all the people that are doing, like you mentioned, the 23andMe, as a bit of a laugh, are probably not cognizant of the potential risks, downside implications of finding out your heritage. It's a cool little party conversation, but it's not going to fundamentally change your life. Is it worth the risk to find out that you are 98% from, I don't know, Iceland?
Yes. Yeah, well, you can. Funny you mention Iceland because they've whole genome sequenced their entire population, and they have probably the best data set in the world for understanding inheritance and disease emergence within a population. So that becomes really useful for research and potential therapeutic intervention.
They also use it for dating, don't they? Just to make sure.
I'm not certain, but there is. I mean, it's a good example. There is.
Maybe that's just a family tree.
Yeah. There's. Yeah. I mean, while it's a little bit of a joke, there is a professor at Harvard, Professor George Church, who was heavily involved in the early CRISPR development. He didn't win the Nobel Prize for it. That was Jennifer Doudna and a French researcher, Emmanuel Charpentier, I think. So, but he was involved in that. Now, he has actually proposed that before anyone starts dating, they should genomically match each other in case they happen to be one of the, 1 or 2% who can't who can't have a viable offspring. So, I mean, it's a very ethically touchy subject. Yeah. His argument is you eradicate a lot of problems before having a potentially non-viable embryo. I mean, you can see where the ethics of all that go.
Definitely.
Yeah. So, but while in smaller populations like Iceland, it has relevance. There are serious suggestions around this type of thing. You just have to be careful that you don't get to the point where you're selecting for certain traits.
Sure.
via the genetic inheritance.
Yeah, I listened to this podcast about a month ago, and this guy and his wife had 8 embryos in storage, and they did a full raft of genetic testing on their embryos, and that informed the order with which they did the implantations.
Right.
So, I mean, it's not they weren't selecting based on specific traits like blue eyes, blonde hair, height, etc., but just on how likely they were to, implant and then go to full term.
Sure.
Which in the back of my mind, I'm thinking, yeah, but surely you've got all the access to the rest of the data behind that. Are you sure that doesn't inform some of your decision making? But it is. It's a very grey area, and there needs to be a discussion around this if more genetic technologies become available and information is more commonplace. Be fun conversation.
Yeah, absolutely. I mean, these things need to be played out, and it's really a public debate that needs to, and an informed public debate that needs to take place, because it's not doctors who are going to have the answers to this. In the end, the problem with fertility medicine is it's incredibly expensive. So you can see where and it's potentially prejudicial against people, against people who can't afford to pay for fertility intervention. So you can see why they might make an economic argument to go with the most viable embryo first. But there are also, I think legally, if an embryo has a BRCA mutation, it may be a reason why, from medico-legally, you may be able to terminate that. I'm not certain of that, but I mean, I don't know the exact law on these things, but you can see where the general public needs to be informed about the potential around what we can predict now. Yeah. And I think that debate hasn't really happened, and I think that there is genomic data getting potentially into the public domain, certainly into the researcher data sets, that needs to be controlled.
Yeah. So why don't we, that's a nice segue to GeneCrypt's mission, which we've talked about, giving the control back to the individual. So you get your genome sequenced, so DNA sequencing, RNA sequencing data, you lock that up so only you have access to it, and you can seed access to at different levels, is that right?
Yep.
So different visibility to different third parties. You mentioned like research or medical. The research thing is quite interesting because I imagine with Medicine 3.0, we're going to need a huge data set. It's not like you can just get one person's DNA or RNA sequencing and be like, haha, I found this exact sequence that is going to inform a lot of decisions in research. You need a big pool, right? So GeneCrypt, this encryption, will really help that. Pooling a lot of data together.
Absolutely. So the value in and particularly as we move towards more machine learning and AI, the requirement to get the enough power into the association is in large data sets or combined data sets into data lakes. And this is what pharmaceutical companies and other researchers want to get their hands on, the large data sets, so they can generate enough power to develop what's called things like polygenic risk scores, because often it's not just one gene, it's a combination of genes or gene variants. So fundamentally, in a genome, you're looking for the variants that person has inherited. You're looking for the variants that are potentially bad, pathogenic, potentially disease-causing. And then there are a whole host of other variants that currently are of unknown consequence. And only with large data sets can you work out what they mean. But with large data sets, you run the risk of identification, and it's impossible to de-anonymize to totally anonymize an individual within a data set. Right. The way that we try and limit that or give control over the re-identification risk is by the individual controlling what parts of their genome are accessed. So the fewer regions of the genome that are accessed, the much less likelihood of any kind of re-identification risk.
Right.
So that's how GeneCrypt does it, by allowing a masking of the entire genome and then only a regional unmasking under private key control. So that's one thing. But the second thing is the ability for data sets from around the world to be combined into one data lake without identification of the individual data subjects, because they're encrypted, and even when the processing takes place with the individual researcher analysis pipelines that people like Sarah and others develop, which are potentially very valuable. They want those analysis pipelines being able to be played out within the data lake without access to what that particular algorithm is. And that's very hard to do if it's encrypted. You have to decrypt a algorithm to run it in a data set, unless you're using something called homomorphic encryption, which is not worth talking about because of the level of compute power that's required to do it. So we've developed a way to carry out that analysis.
Right, so it's encrypted on behalf of the individual, so that's entirely masked.
Yes.
Anyone looking at the data set would never be able to connect a data set of an individual with an individual.
Yes.
That's great. And then researchers, they might have intellectual property around how they mine the data, and that also is encrypted. So you can have concurrent access by multiple different researchers running different investigational algorithms, and they have no idea what those algorithms are from each other.
Absolutely.
Incredible.
I want to take that soundbite away, because that explains it a lot better than I can.
No, that's awesome, because we learn constantly that whenever you're doing something with a lot of numbers, whether it be statistics or some kind of mathematical algorithm, whatever, it's junk in, junk out, and often the junk going in is because you have a small data set. It's not powerful enough, and you're going to get a whole bunch of errors in it. If you have a small sample size, the error rate is very high, because who knows, based on sampling bias, you've got completely unrepresentative data in your data set. But when you magnify, I love the phrase data lake.
Yes.
I've never heard that before, but it's so good. You've got little pools that add up to a lake.
Yes.
You've got this big, deep, wide lake of data, much more powerful.
Yes.
Absolutely incredible. So why don't we talk about?
Just on that point, it is. I mean, you're picking up on the real value in it, because because researchers, big pharma. I mean, this is how you identify, because we still don't know a lot of the effect of the genome. So, and as Sarah's researching, a lot of that junk DNA, or the long non-coding RNA. In the dark regions of the DNA are not understood yet, and there's 98% of it, as I understand, are in the dark region, and we don't know a lot about how that works. So it's really important that the potential value of those, either whether they're for good or bad, that we understand what they are before that data is released. Secondly, big pharma or researchers or whoever it is, and the value should be back to the individual, I believe, and if their data is being used, there should be some value back to the individual. But they want to tie up we talked before about biomarker data or Phenomic, that your outcome data, how old you are, what diseases you might have at the moment, what your blood pressure is, what your cholesterol levels, all those things. They need those data in these data lakes to do the association studies. And those are highly identifiable. So you need a way to conduct those studies in a way that you've masked out connectedness between them.
Yeah. Man, so cool. I mean, I think we take for granted, or we don't acknowledge how complex all of this is and how much information is really required to do really high value, high level investigation. Dr. Sarah Diermeyer, who you mentioned, she talks about how 2% of DNA is coding for proteins. The central dogma of the human body is that DNA, mRNA, protein, function. Very linear, very simplistic, but it's a good way to teach, high school. But in reality, it's infinitely more complex. And if anyone has seen, like proteomic maps of all the interactions between proteins, I mean, it's just a mess. Yes. And then you don't just have protein. You've also got the RNA, all the different types, the link RNA that Sarah's doing. That we're finding that it's more and more implicated in a lot of different things. So unless we have sufficiently powered data sets, we're not going to get good data out of it. And if it comes back to the importance of the individual, they need to get value out of it. So if it's done poorly with a set of 5, that's the outcome of that is not worth the risk to them. So I think that if I'm understanding correctly, GeneCrypt provides both encryption, sufficient power for investigation, that both of those together, you can't have them separately, but both of those together provide the value back to the individual, over and above just knowing their personalized data.
Absolutely. There's a step beyond that too. I mean, you've raised the point about proteomics around metabolomics, I think it is. And so we're getting into the age of multi-omics, including even things like I think it's radiomics, which is the radiological data, and trying to link that with pathological data with genomic data. So there's very much we're building up these association studies and trying to, and this is where the power of AI comes in, because it's so complex that you need those AI algorithms to try and work out what the associations are between them all. But I just don't think that we haven't developed the infrastructure yet to contain that data and also to share it. I mean, we need to think about these things, potentially decades ahead. This is, an example in New Zealand is when I was working in the health system here, we were talking about, and I'm going back a decade to trying to get clinical pathway platforms in place that still aren't there, to try and standardise how someone's treated when they come into a hospital. But, we're still talking about, I think this is about the third or fourth iteration of trying to get a national data set together for healthcare. Currently, I'm working with a researcher and a cancer surgeon, Simone Petrich, who in the South Island, University of Otago, connected in Southland, DHB, they're trying to get a familial ovarian breast cancer database together, so we can at least try and understand the inheritant or the types of patterns of genomic and pathological inheritance. New Zealand has a very high rate of breast cancer. Yep. We don't know why that is, and we don't know what the genomic patterns of that pathology are. So there is work at play trying to put in or trying to at least start with some disease registries, and then increasingly we can build the genomic data into those. But we need to be thinking, 10 and 20 years ahead, so that we can start getting the basics in place, because we're still not even doing medicine 2.0 right. I mean, and this is not a commentary on any one of the parties, but I think New Zealand, on the whole, as a health system, have failed to look 10 or 20 years into the future. And we can get onto it, but I mean, I'm helping. I'm a mentor for the chiasma program in New Zealand, the STEM students at universities, that's science, technology, engineering and maths. And we've got some really top quality, graduate and postgraduate students coming through in STEM, but we need to bring them into the problem solving for the future of the New Zealand health system. And genomics is just one part of that.
Definitely. That's an interesting topic. I mean, how you, obviously this is, you, like you're saying, you're thinking 10, 20 years into the future. How easy or difficult has that been to convince people to start thinking about these things now? You mentioned that healthcare system doesn't really do that. They're kind of thinking of the here and now, and struggle to adopt some of the best practices that are in the world currently under Medicine 2.0. But what has that been like? Has that been a struggle?
Yeah, very difficult, extremely difficult in New Zealand. I think part of the problem is, as an economy, we are still focused on domestic housing as a consumption item that everyone is trying to sell on an increased basis to one another. We're focused on Fonterra and dairy technology, and selling. And we've actually sold the technology to other countries to compete with us in the primary industries, and we've got tourism. So those aren't long-term sustainable, export-driven. I mean, you could argue milk's export-driven, but it's
not high value.
It's not high tech. It's not high value. We need to start thinking really deeply about biotech and deep tech, and the future of the world, because that's where it's going. And if we want to do that, we need to start enhancing the kind of risk capital for startups. And there really isn't a mechanism in New Zealand for developing biotech and deep tech. Certainly at the startup level, you can do it in universities, but if you're a startup founder, you just won't have any funding here to do it. There's virtually zero government funding for startups at that level. I mean, Callaghan, you could argue, deal with maybe corporates or people who have at least 300,000 plus of resourcing, but we need to develop a mechanism for those capital flows, or particularly risk capital, because it is risky, particularly in the really early stages of development and startups, particularly in bio and biotech. So we need to build that infrastructure within New Zealand, and we've got really good people coming through in our STEM programs, but we need to build a place for them to go in New Zealand without them disappearing. And I think to do that, we fundamentally need to move away from this belief that buying a rental property is the way to become a multi-millionaire, because it has been, because all the politicians all own 3 or 4 rental properties. Perhaps if politicians were forced to have equity in a biotech or a deep tech startup, we might have a different economy and a different focus over the next 10 to 20 years. But it is a long-term thing. It can't be an election issue. It can't be a short-term nice little soundbite facade. It needs to be a multi-decade provisioning.
Because that's the difficulty that we have, isn't it? Fundamentally, it's very expensive, very risky, and the time horizons are generally a lot longer than in other domains. I mean, if you have a culture that's built around capital gains of housing, tourism operation, and farming, the time horizon is generally fairly short by comparison. I mean, maybe 10 years ago, you could have bought a house and in 3 years made quite a lot of money if you'd played your cards right. If you're a farmer, you can make money that season or the next season. Not quite as short when you're dealing with biotechnology. So maybe it's a cultural ingrainment that we have of shorter term time horizons, and the risk appetite just isn't built into that. I mean, we do have a few venture capital firms based in New Zealand that do a little bit of this. Right?
Yeah, very, very. I mean, if they'd like to get in contact with me, I'd love to talk to them. They're incredibly difficult to gain traction with. This is the dilemma for founders is you get given a lot of specious advice, often well-meaning, and they might they have programmatic approaches to these things. Get a great idea. Find a large addressable market. Work up the unit economics from the ground upwards. Build a kind of working model with a front end. Then you go back and see them. They say, well, can you build a prototype? So you build the prototype. You go back and see them. Well, have you got any traction? Well, you go get some traction. You go back and see them. No, you're kind of too early for us. Have you got any clients? Have you got any paying clients? So you go back and see them. You've got a paying client. Well, are you profit generating? So at the point that a VC in New Zealand, I mean, it's different in the US. I've been in the US and spoken to a number of them. They have a much different approach to it. In New Zealand, the VCs here pretty much won't invest in you unless you've got a lead investor or significant family, friends backing. And we're talking into the hundreds of thousands, and you've used that risk capital to de-risk it to the point whereby you've got paying customers, and at that point, it's arguable whether or not venture capital is always good at any point, but they've de-risked it so much at that point that it's almost useless.
I see. Yeah. So who needs to fill that vacuum?
So the founders have most of them will have disappeared by then. Yeah. And the problem in New Zealand is you've got a number of these entities that are well-meaning and well-meaning, but very few of them have the deep kind of domain expertise you need, or indeed founder experience. Lived founder experience, and you can read all the books you want, but you've got to understand it at a gut level. Because it's survival of the fittest, and not many survive.
Absolutely. I mean, from my perspective, there's quite a large number of risks that I think New Zealand, the ecosystem, does face. I think you raise a really good point, that gap between actually getting outside capital into the company via VC and not having huge government support or government entity like Callaghan support compared to maybe our brothers and sisters in Australia.
Yes.
They get a little slightly different arrangement over there. And then there's also, we hear a lot about brain drain. If there's nothing here for people, they get trained up and then they go. So it's a huge expense to educate them, and then they take that knowledge elsewhere. I mean, I'm a classic example of that. I live here, but all of my employers are outside of the country. I mean, they're headquartered outside of the country and employ me via third-party employer of record. So, that's part of the reason why I'm doing this podcast, because we need to have a dialogue around. I mean, we're all fairly patriotic, but there comes a point where it's not going to be sustainable and we will have missed the boat. So if we don't talk about it now, don't talk about it in the next 5 years, it might be a different story.
Yeah, absolutely. And it's critical for those students going through STEM programs at the moment, who are going to be the resource that we need to build these either startups or feed into the once startups have become significantly revenue generating and become employers within New Zealand. We need a way to make it more secure you'll never make it secure for founders. There are much more there are much better founder-friendly economies in the world whereby there is a lot more allocation of risk capital, and that risk is understood by more sophisticated investors and angels and all that kind of thing. We have some angel groups here, but typically they're not going to pick anyone up at a pre-seed stage. They're not going to provide significant seed funding. And so that's where I see that gap. There's another problem for founders and startups, and it is you often burn your resource up trying to get the prototype and traction, and you don't think ahead to the next battle. So you've burnt you get told, go get some traction. You burn all your resource in that prototype, and then they want paying customers, or they want the UI on the front, or they want the full stack with the platform deployment, and you've burnt your capital. It's kind of like a battlefield commander. You need to start thinking about the next battle, not the current one.
Yeah. Have a war approach as opposed to an individual battle approach.
Yes.
Yeah.
I mean, I think often some of the military analogies and the Sun Tzu and maneuverist approaches are probably quite relevant, perhaps more relevant than MBA formal training. I think being able to fight battles, be resilient, be resourceful, and be persistent are probably skill sets that are required in a startup world.
Definitely. Well, Kris, this has been super insightful. Thanks so much for your time. Really appreciate it. And I'll be watching very closely as to what happens with GeneCrypt. It's very interesting. Really looking forward to seeing what other researchers get on board to make use of your technology. Where can people go to find out more about yourself, GeneCrypt, and where the company's heading?
Sure. Well, we have, if you just look up GeneCrypt in the news, you'll see the press releases that have come out in the US. We're currently trying to make headway into the US because the North American market is half of the world market. We've got a website, GeneCrypt.io. That's GENE Crypt, which is CRYPT.io. And I'm on 021 889-204. 021-889-204. Anyone can contact me through my mobile. That's a New Zealand number, so it's +64 on the front, and or through the website. And we are under FMA rules. We're not allowed to advertise to the general public, but it's just a general known rule that every founder's always looking for funding, always looking for partners and connected relationships. So please get in touch if you're interested in any way or form with genomics and genomic medicine.
Awesome. Thanks, Kris.
Great. Thanks,
All the best.
Charles. It's been good.